Hello. Hello. Am I audible?
No,
Please speak up. Am I audible? Hello. Hello. ওইটা অন এই মাইকটি হ্যালো 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 এই সামনে তো জন্য স্যার একসাথে হ্যাঁ একটু থাক ঠিক আছে হ্যালো হ্যালো
Hello.
We need the connection to be checked. Can any of our participants please speak out? This is Dr. Himang Sumati from JISU University. Hello.
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 Hello, sir.
Hello.
Hello. Am I audible, sir?
Yes, sir.
Sir, am I audible?
Yes, yes. Hello, Sakhar.
Yes, sir.
Can you hear me?
Yes, sir. I can hear you. Yes, sir. I can hear you.
Your sound is not coming.
Yes, sir. I can hear you. Can you hear me?
A little bit louder, please.
Yes, sir. I can hear you. Can you hear me?
Your sound is actually not coming.
Hello, sir. Can you hear me?
Yeah, I
Hello?
can hear you, but the sound is bare minimum. Speaker volume.
No, done. Select the speaker. Which one?
No. There's same as system. Hello. Yeah, I can see that Mr. Soman has opened the mic.
Yeah, I mean,
Can you start the program?
Hello, sir.
Yes.
hi.
Audible properly, sir?
You are audible.
Yeah, I can hear you.
Okay,
Can you
sir.
hear
Okay,
me?
sir.
Start, na?
Can you hear me?
Yes, I can. So,
Okay.
thank you. So we'll start very soon. Fine, no? So, ল্যাপটপের ভিডিওটা নয় দুটো ভিডিও হ্যাঁ এটা অফ সাউন্ড চেক নিয়ে না বড় কিছু আছে তো আছে ঠিক আছে দুটো লাগিয়ে
সরি না কথা দুটো আছে কিন্তু আমি স্ক্রিনের মধ্যে স্ক্রিন
আমারটা
ওইটা নিচ্ছে
আমার ক্যামেরাটা বন্ধ
আমাদের ওটা বন্ধ করতে পারবে না কাজ করতে এর উপর ঘুরিয়ে আর ওই তো ওই আলোটার জন্য প্রবলেম
ওই আজকেটা বন্ধ করেছে
এক মিনিট ওভাবে গাড়ি দাও সব গাড়ি ওইখানটা বিরাট ऊपर लैपटॉप में चार्ज नहीं चार्ज पे है ना चार्ज ठीक है ना ना आई थिंक এবার তুমি ক্যামেরাটা চালাও ঠিক এটা অনেক বেটার ঠিক আছে আরেকটা কাজ করতে হবে আরেকটু যদি পিছিয়ে নাও আরেকটু ওরকম না ঠিক আছে চল
Good afternoon, everyone. It gives me immense pleasure to extend to you a very warm welcome on behalf of the Department of Pharmaceutical Technology, JIS University, and Internal Quality Assurance Cell, JIS University, Kolkata, who has clubbed all of us for the 5 days faculty development program on post-pandemic advancement in drug research and sustainable development. Myself, Dr. H. Assistant Professor, JIS University.
and I, Mr. Sakshar Shah, Assistant Professor from the Department of JIS University. So, before we start our program, I would like to quote quotation from a renowned academician. He said, "The aim of the education is the knowledge not of fact, but of values." It is on the occasion like this we get the opportunities to test our knowledge and the understanding we did develop. We look forward to get an exposure about what is the best of the human brain. Think about very dynamic issue of JIS University, to be specific, the Department of Pharmaceutical Technology, JIS University.
JIS University was established through the JIS University Act 2014 and enacted by the Legislative Act of the Government of West Bengal that came into force in February 2015. The university's objective is to rank among the best institutions of higher learning in India and to be a top choice for teachers, researchers, and students. All courses at JIS University have been properly accredited and affiliated by the University Grants Commission, New Delhi, the All India Council for Technical Education, the Bar Council of India, the Pharmacy Council of India, the National Council for Teacher Education, and the United Nations Academic Impact. This guarantees prospective students a high-quality education. Since 2017, JIS University has also belonged to the Association of Indian Universities. The Department of Pharmaceutical Technology of JIS University started functioning with Bachelor in Pharmacy with prior approval from Pharmacy Council of India, New Delhi. Consequently, affiliation from AICTE was obtained for pharmacy program. Currently, we offer Diploma in Pharmacy, Bachelor of Pharmacy, and Master of Pharmacy in Pharmaceutics, Pharmaceutical Chemistry, Pharmacology, and Quality Assurance. We also offer PhD in Pharmacy.
The COVID-19 pandemic response has engaged the academic, public, private, and health sectors in the real-time development of technologies, practices to enable predictive, preventive, personalized, and practitionary health. We all have faced the situation, and we all know that how the pandemic situation the health sector has been through. Myriad cases of collaborative innovation across various sectors have emerged throughout the pandemic response that serve as example to address post-pandemic health system challenges. Despite of certain observed technical, social, institutional barriers, the collaboration should be carried forward to entertain post-pandemic advancement in drug research and development. This FDP program sets out a series of research priorities that could inform interdisciplinary collaboration between the clinical sciences, public health, technology, healthcare providers, and others. The latest development in the therapeutic research will be discussed in this session.
Music heals, music entertains, music takes us to heaven. Our music team is on the stage with a beautiful song to welcome our guests, delegates, and dignitaries. Now I call upon the stage the members of our musical team.
আলোয় আলোয় এই আকাশ
Thank you so much for this lovely music, and to start this program with a beautiful music, I think always is a blessing. Now, how about our classical dance? We all have a affinity. We all have an affinity towards our cultural tradition. Our ancestors used different forms of art to tell the stories and convey their feelings and emotions. Now we have one of our students from MPharm Pharmacology to present a dance, or you can say to tell a story, to entertain us with her dance. Her name is, she is from MPharm Pharmacology. So let me welcome her through this program. She is from MPharm Pharmacology. So let me welcome her through.
मधुकैटव विध्वंसी विधात्री वरदे नमः रूपं देहि जयं देहि यशो देहि दिशो जहि महिषासुरनिर्णाशि भक्तानां सुखदे नमः रूपं देहि जयं देहि यशो देहि दिशो जहि धूम्रनेत्रवधे देवी धर्म कामार्थदायित्री रूपं देहि जयं देहि यशो देहि दिशो जहि रक्तबीज वधे देहि चण्डमुण्ड विनाशी रूपं देहि जयं देहि यशो देहि दिशो जहि शुम्भ शुम्भ निर्णाशि शुभदे नमः धर्म कामार्थदायित्री रूपं देहि जयं यशो देहि दिशो जहि रक्तबीज सर्वसौभाग्यदायिनी चण्डमुण्ड विनाशि रूपं देहि अचिन्त्य दिशो चरिते सर्वशत्रुविनाशिनी रूपं देहि जयं देहि यशो देहि दिशो जहि न तेभ्यः सर्वदा भक्ता दुरितापहे रूपं देहि जयं देहि यशो देहि दिशो जहि सावज्ञो भवति पूर्वं चण्डी जगन्नाशिनी सर्वशत्रुं विनाशिनी रूपं यशो देहि चण्डी सततं जयन्ती स्वामिनाशिनी
I think I'm audible to all of you. Am I audible? If anybody respond kindly. Am I audible to all of you? Am I audible to all of you?
Yes, audible.
Okay, thank you so much. So thank you so much for being with us. We all know we are a little bit not so sound with the technology. So sometimes we are audible, sometimes we are not. And thank you so much for being with us with with this kind of difficulties and inconvenience. So I would like to thank our MPharm second year student Vidisha Das for her very awesome performance and entertaining us.
It is a great pleasure to mention that our department is taking an initiative to publish a scientific peer-reviewed journal named the Journal of Integrative Science and Drug Research. Professor H.S. Maji is the editor-in-chief of this journal, and Sheikh Abdul Amin is the co-editor.
JISDR, that is Journal of Integrative Science and Drug Research, it is dedicated to studies on the main aspect of pharmaceutical, biological, and biomedical sciences. It provides the medium for publication of original research paper and also welcomes the critical review papers, including short communications and others. The Journal of Integrative Science and Drug Research, the editor-in-chief, as my co-host said, Professor H.S. Maji, and co-editor Sheikh Abdul Amin. We work as a team and aims to publish quality papers or publication or article that are reviewed in an unbiased manner by editorial and review board members and make them freely available to the researcher worldwide. It is a small practice from the JISU University Department of Pharmaceutical Technology to help you to publish your research article in a journal made by us.
Now, I would like to request Professor Himanshu-Shekhar Maji, Dean Academics, JIS University, to deliver the welcome address.
Sir.
Yeah.
Yes, audible.
Good afternoon. I hope I am audible to all of you.
Good afternoon, sir. Yes, you are audible.
Yeah. So, on behalf of the Department of Pharmaceutical Technology, JIS University, I would like to welcome all the registered participants, faculty members of different colleges and universities in our faculty development program. This is a kind of second faculty development program that we are offering from the Department of Pharmacy. This time the theme of the program is post-pandemic advancement in drug research and sustainable development. Now, faculty development program is essential nowadays for a faculty members whose experience is 0 to 5 years. This is a kind of career advancement scheme also, where we used to discuss the different pedagogical techniques and other attributes required to be a perfect teacher in any educational institutions. So I'll share a few points regarding the development of your own faculty when you are trying to establish yourself as a perfect teacher. Now, this is not my word. This has been coined by NITTR. Perhaps many of you have undergone the modules. It contains 8 modules. It says that the objective, the first point is the orientation of a new faculty members towards the academic ambience where he or she is teaching. And also he must be oriented towards the curriculum and educational techniques. The second is a faculty member must be sufficiently knowledgeable regarding the professional ethics and sustainability into an academic environment, into a college or universities. We must focus also on the communication skills. Each and every faculty member should focus on his or her ability to deliver the technical knowledge to the students. The dissemination of the knowledge becomes perfect when a faculty member tries to communicate and is perfectly able to communicate what he or she is thinking to provide. So, in terms of communications, we must be a little bit straightforward. See, communication sometimes we do misinterpret with the spoken ability into a foreign language or say English so far. The communication, there is a little bit difference between the communication and English. Communication is the way you are expressing your ideas to the students. You must communicate to each and every participants in your class. You must relate so that 100% of the students can relate yourself whatever you are delivering in a 45 minutes or 50 minutes class. So this is known as communication. Sometimes it requires say definitely English, but many times it requires other teaching aids like say PowerPoint presentations, animations, etc., etc. So communication is a major thing a teacher or newly adopted teacher must focus on. Then a teacher also must be very much focused on the planning and delivery of the content. While going to the class as a newly inducted teacher, you must work sufficiently so that whatever you are going to deliver in the class, you have a rehearsal prior to entering into the class. So that planning and delivery must be perfect before entering into the class. Then we must also use the technology-enabled learning and lifelong self-learning pedagogical techniques. Nowadays, teaching learning is not limited within the blackboard and chalk. With the advent of computers and PowerPoint presentations and animations, and also with the addition of internet, teachers nowadays is only a medium to communicate all the knowledge to the students. So teacher's role has now been redefined from the content delivery person to nowadays in many of the schools it's called teachers are not teachers, teachers are facilitators. We do not deliver new things, rather we assist our students to gather the knowledge from the different sources. So teachers can be like an encyclopedia. So you must have sufficient knowledge about the resource where the study materials are available. Then a particular faculty members, newly inducted faculty members must know regarding the student assessment and evaluation techniques of a university or college is offering. So this assessment and this examination or evaluation of the students varies from college to college or university to university, but most of the time the major objective is to know how a student understands a teacher's delivery, how he gathers knowledge. So the examination pattern of a university or college, a teacher must be well aware. Then also, apart from only giving knowledge or say PowerPoint presentation, a teacher must be interactive in the classes. He must interact with every students or almost all the students so that the teaching learning process can become more participative in terms of creative problem solving, innovations, and some kind of research mindedness must be imparted to the students. So it is the role of the newly inducted teachers to get themselves acquainted with the pedagogy, with the research motives, with the innovativeness that you are giving to the students. Finally, after gathering all these things, a teacher must also adopt the institutional management and administrative procedures. Yeah, teacher's role is nowadays not limited to teaching learning process, but the teachers are doing so many things nowadays that out of say 30, 35 or 40 odd jobs, teaching is one kind of job. So you have to do everything. Sometimes it is it is expressed that teacher is doing everything except teaching. No, my dear friends, you keep teaching as the top priority, but side by side you have to do a lot of work regarding the administration running of your academic institutions. So this faculty development program helps us to refresh our knowledge, to refresh our teaching pedagogy, to refresh ourselves so that we can deliver the best to our students. The objective is to increase the knowledge and skills of the students. We from the Department of Pharmaceutical Technology, JISU University, is taking this kind of initiatives, and we hope that we can add some value to your academic credentials at the end of these 4 days. Now, in this program, we have a number of speakers to whom you will be interacted with. We have Dr. Somen Kanti Manna immediately after my speech. He is an associate professor of biophysics and structural genomics, division of Shah Institute of Nuclear Physics. He will be discussing today with metabolomics applications and in mechanistic elucidation, drug repurposing and personalized medicines. Drug repurposing nowadays is most widely used term, at least after this COVID pandemic. Then we have speakers like Professor Umesh Kumar Patil, Mr. Ashit Kumar Lohari, Dr. Girisha T. Mohanna, Dr. Chinmoy Patro, then Dr. Tapan Kumar Giri, Dr. Animesh Ghosh, and Professor Achinto Kumar Saha. They are from Calcutta University, Birla Institute of Technology, Mesra, then Jadavpur University, and Dr. Patro is scientist E from Division of Developmental Biology, Pune. So we hope that this 5 days will going to be a successful one. I urge you to interact with our speakers and participate in all the sessions so that our report becomes useful. Thank you. Thanks a lot.
Thank you so much, sir, for your thought-provoking address, which sets a platform for this event, for this FDP.
Now, I would like to request Professor Kamala Kanto Roy, JIS University, to introduce the FDP coordinators and also to share the rules and regulations of the FDP.
So thank you, Devjani and Saksa. So each and every event, to conduct each and every event, there are some persons who are dedicatedly engaged for smooth running of the program. So we also get some of them as coordinators and joint coordinators. Mr. Dr. Arinda Maifi, Dr. Ritu Khara, and Sheikh Abdul Amin. So Dr. Arindam Maiti is an associate professor at the Department of Pharmaceutical Technology, JISU University, Kolkata, India. He was awarded PhD from Jadavpur University, Kolkata, India. Also did his research for postdoctoral program from University of Southern Denmark. He has published 55 research review articles in different reputed peer-reviewed journals. And Dr. Ritu Khara is an associate professor at the Department of Pharmaceutical Technology, JISU University, Kolkata, India. She was awarded PhD from Jadavpur University, Kolkata, India. She has 7 years of teaching experience. Research includes synthesis of small molecules with anti-cancer and antiviral properties. Dr. Khara has published 25 research reviews articles in different peer-reviewed journals and 4 books chapters. And Sheikh Abdul Amin, the youngest scientist, is an assistant professor at the Department of Pharmaceutical Technology, JISU University, Kolkata, India. He has submitted his PhD thesis at Jadavpur University, Kolkata, India, and he has been placed among the top 2% of scientific achievers worldwide in single year 2019 and 2020. His research area includes design and synthesis of small molecules with anti-cancer and antiviral properties. So thank you all the coordinators and grant coordinators. Hope the FDP program, 5 days FDP program will be successfully managed by these coordinators and joint coordinators. Thank you.
Thank you so much, sir, for addressing the coordinators of this FDP program. Now, I would like to request Dr. Ritu Khara to come here and introduce our eminent speaker of today.
Dr. Shoman Kanti.
Can you hear me?
Yes, sir, you are audible.
Oh,
Okay, good afternoon to all. Myself, Dr. Ritu Khara, Assistant Professor, JISU University. I would like to welcome you all in the first session of FDP entitled Post-Pandemic and Advancement in Drug Research and Sustainable Development, organized by JISU University. It is my great pleasure to introduce our eminent speaker, Dr. Shoman Kanti Manna. Dr. Manna is an outstanding researcher and eminent educationalist. Dr. Manna received his bachelor's and master's degree in chemistry from University of Calcutta, India. He received his PhD in chemical sciences from Tata Institute of Fundamental Research, Mumbai, working on elucidation of structure, function, relationship, and engineering of a microbial cytochrome P450. He did his postdoctoral research at the National Cancer Institute, United States of America, on identification of biomarkers of exposure and pathogenesis, as well as elucidation of the underlying mechanism. Currently, he is an associate professor at the Biophysics and Structural Genomics Division of Saha Institute of Nuclear Physics, Kolkata. His lab is working on elucidation of metabolomic reprogramming associated with stress response in cancer and liver diseases, and the role of RNA modification in stress response. He takes interest in method development for chromatographic and mass spectrometric analysis for small molecules. His lab has developed a cost-effective non-enzymatic method for simultaneous analysis of several DNA and RNA modifications. He has contributed to identification of 4 novel metabolites. His lab is also involved in identification of diagnostic and therapeutic biomarkers of metabolic syndrome and respiratory diseases using metabolomic, lipidomic, and peptidomic analysis. His work has been recognized by patents and awards including NCI Directors Intramural Innovation Award and Ramanus Fellowship. He is enthusiastic about promotion of metabolomics and his conduct 4 workshops in mass spectrometry and metabolomics in his lab. He is quite actively involved in promotion of science and education in schools and colleges in remote and underprivileged area. This is very short and very brief introduction about our speaker. Now I would request Dr. Mana to deliver his speech. Sir, the stage is yours.
Okay, thanks, Ritu, and thanks to all the organizers for this opportunity. It's my pleasure to share a bit about metabolomics. Am I audible? Am I audible? Clear?
You are audible, sir.
Yes, you are audible, sir.
Switch on your video.
Okay.
Yes, audible, sir.
Okay. Okay. So let me continue then. So I mean, do you really need the video? I can turn the video on for a while because I think at times, the connectivity issues pop up, and I'd rather like you people to listen to what I'm saying rather than to look at what, how I look like. So, okay, fine. Okay, so I'm going to share the slide and let me know when it is visible. I hope it is visible now. Is it visible? The slides are visible?
Not yet, sir.
Not yet? Okay, just let me give the one more. Okay, how about now?
So now you can share your screen, sir. If you can try once.
Okay, is it visible now, the slide?
No, sir.
Not yet?
No, sir. It is not audible. It is not visible.
Okay.
Sir, in the Zoom page, if you just are you like clicking the share screen option, which is in the green button?
Yeah. That's what I did. Just 1 second. I will try it once more. Okay.
Okay, sir. Okay, sir.
Okay.
Yes, sir. It is coming.
Okay.
Yes.
Okay. I hope this will work now.
Yes, sir.
Okay.
Yes, sir. It is visible, sir.
Okay. Thank you. So, okay, let's get started. So as you see from the title, we would like to talk about metabolomics and its applications in mechanistic elucidation of gene environment interaction or pathogenesis. And we'll talk about a bit of its application in drug discovery and drug repurposing, as well as personalized medicine. So as you can see, this is quite a big spectrum. So it will not be possible to cover every corner of it. So we would like to, I'd like to have, some glimpse of, each of these so that you get a flavor, and I will obviously be available for further discussion, both after the talk and beyond. Okay. So let's start with what is metabolomics. So if you look into the history, this is kind of old wine in new bottle. I'll tell you why. As you can see from this picture, which is taken from Jeremy Nicholson's paper, so this is called urine wheel, which was published by Ulrich Binder in his book in 1506. That's quite a while back. And in this picture, you can see these are basically, bottles of urine. And looking at their color or smell or the density, etc., people used to guess what kind of diseased person may have, or what kind of physiological status that person may have. Now, as we know, for several inborn errors of metabolism like apple syrup disease and stuff like that, the urine density, for example, as well as color, really changes. And we also know, for example, in several conditions, including infectious diseases, the urine smell itself can change. So therefore, the concept that some of the biofluids may have something in them that may be reflecting the health of a person is something not new. What is relatively new is that those days people didn't know that what is that causing those changes in either physical appearance or the smell or whatever it is. Okay, so that's where the new thing is coming. I'll tell you why this resurgence of interest in metabolomics is there. So this term itself was coined sometime in 1998, and this refers to the quote unquote definition of metabolomics is basically global analysis of metabolite biochemicals, basically small molecules which are less than something like 1200 Dalton, and what is that they are here for. Okay, so that's what metabolomics means. So generally, you will analyze this, and then you will identify and measure the concentration of the metabolite in your matrix of interest. That can be cell, tissue, biofluid, or it can even be some sort of environmental sample. Generally, when you do these measurements, you would like to connect it to a particular kind of state, a perturbation that you may have caused or that may have already been there. So this is either an endogenous perturbation or an exogenous perturbation. It may be a pathological state, a disease, or it can be exposure to something, etc., or it can be even a mutation inside. This is an endogenous perturbation. So in connection with that perturbation, you would like to then understand that how this biochemical fingerprint has changed. And in many cases, the change in this biochemical fingerprint can be quite a signature and can be used as a biomarker for that particular condition or exposure. Okay, just to give you an example, we actually identified metabolites which are signatures associated with radiation exposure, ionizing radiation exposure like gamma ray exposure. Okay, so it can be as simple as that. And of course, we have biomarkers for diseases, and some of which we will discuss. So then you would like to characterize the biochemical phenotype not only by measuring and identifying compounds, but also how they are connected to each other. From there, you would like to develop a hypothesis that how this change has been arrived at. And then you will test that hypothesis, then validate that, and eventually you get a mechanism. And only when you have a mechanism, you can think of some ways to modulate the phenotype, okay, or to manipulate the condition that that person may be suffering from, or that whatever it is. And this is where therapy comes into picture. So therefore, all the way starting from analysis of the metabolites to the mechanism, you have to traverse the whole way in order to identify the mechanism. Mechanism-based therapeutic target, which then can be, the drug development can kick in. Okay, so I will try to give you during the course of the talk, some of the flavor of each of these aspects, including biomarkers, mechanism, and also how that it is, the patient stratification itself can take place, and in the context of personalized medicine. Okay, so yeah, so then you can have design methods. And now, why metabolomics? So if you remember that, the Human Genome Project was conceived with the hope that once we have read the genome, we will really know how a person is, whether that person will be fat or lean, whether the person will have a disease or not. Hello? Can you hear me? Is everything okay?
Yes, sir. Everything okay.
Okay. There is some noise in the background. Okay. So so with that kind of hope, it all started, and even after the completion of the, sorry, there is quite a bit of noise in the background. So I think somebody is speaking.
No, sir.
Maybe so. Anyway, so people may kindly switch off their mind because I get feedback. So anyway, so that didn't really happen till date. We have only 10% of the global disease burden that can be explained really with the genomic cues. But if you see, this is the the flow of biological information, that things may start from gene, but then they eventually go to mRNA, then to protein, and then to and the function of the protein, some of which can be enzyme producing metabolite, etc. But that's your endogenous machinery. We are never in isolation, so we always interact with the environment. So this gene and environment interaction is something what gives you the phenotype. Okay, so finally leads to function and phenotype. For example, you are listening to my talk, and I'm not changing your genome. I'm not doing any physical perturbation to you. Like listening to someone, or even thinking of meeting or listening to someone itself, can be quite exciting or quite depressing, depending on what the person is, okay, to you. So those are the type of interaction or perturbations that are not even physical or biochemical or anything. These are some virtual interactions or virtual perturbations. And if you see, eventually things that change your mood, etc., is a chemical. So everything inside our body is perceived chemically and executed chemically. So this is where both the environmental cues and endogenous cues, when coincide together, eventually to give something which is a chemical output. And this is where the metabolome is extremely important, because in the metabolome, you are looking at the biochemical landscape, you are trying to read the behavior of the biochemical machinery. So metabolome, in a sense, is the ultimate outcome of the upstream intertwined biological information cascade, which is the closest to the phenotype. And this is where we would like to start, so that we get the inputs from both environment as well as from the genome, as well as their interaction. So, and then we can be in a better position to decipher the mechanism and to understand what's going on or what's going on. So this is why metabolomics is important. It's the closest biochemical representation of the phenotype that you are looking at. Now, what is important to look at, just to give you an example, if you look at a genome, if you are thinking of a polymorphism or a mutation, then it can either be there or it may not be there. So that means it is binary, 1 or 0. So if you calculate the fold change, as we generally often talk in biology in terms of fold change, like gene expression or protein expression, that is an infinite fold change. If you think of the fold changes that we often observe in the transcriptome, etc., can be 5-fold, 10-fold, etc. But think of something as simple as your blood glucose level. If your blood glucose level, fasting of course, is less than 100, then you are apparently normal. If it is consistently more than 150, then you are probably diabetic. So this is just 1.5-fold change in some biochemical. That is telling you that you have a disease, and so many redundant underlying mechanisms and pathways have already failed, and that have resulted just 1.5-fold. Think of, if you have a 5-fold increase or decrease in sugar, you will just perish. So this is where all the canonical pathways actually, coalesce together in order to tightly regulate the biochemical homeostasis. And this is why it is extremely important to note even the minuscule changes that you may have in your metabolome, and that may be quite significant. So to say that as you start from genome, go all the way to metabolome, generally the fold changes or the magnitude of changes are lower and lower. So there is an orchestrated dampening of change. And also one has to remember that metabolome particularly can be affected by several things. Can be affected by your food, your pesticides, the drugs you take, pollutants, the cosmetics that you use, and several other things. And on top of that, you have the gut microbiome, and eventually the output is metabolome. So therefore, trying to look for a metabolic fingerprint or metabolic signature associated with a particular condition is pretty much trying to look for a needle in the haystack. It's quite an involving process, but it's quite a fascinating journey as well, because deep underneath that little signature, that little change in signature has actually got all the information which has latent signatures of the upstream biological cascade. So that's why, although it is difficult and challenging, we would definitely like to understand what it is. So this is a typical workflow of a metabolomic experiment. So you start with some sort of biological sample that can be cell culture or cells, isolated from some living organism or a tissue or a biofluid. And then you have to extract the metabolites. And in some cases, you may not actually need to extract metabolites. For example, if you are directly doing something called metabolic imaging using mass spectrometry, you do not need to extract anything. Or even in NMR, you have very little preparation that is involved. And then
for the kind attention of the passengers, train number 38824 Medinipur Howrah local is arriving on platform number 8 অনুগ্রহ করে শুনবেন গাড়ির সংখ্যা 3882 মেদিনীপুর হাওড়া লোকাল 8
please someone
All the participants are requested to please put yourselves in the
মেদিনীপুর হাওড়া লোকাল প্ল্যাটফর্ম ক্রমাঙ্ক
all the participants are requested to put yourself in the mute mode, or if you're outside, kindly you can leave the meeting, because our eminent speaker is on the middle of his presentation, so it is making a little bit inconvenience for all of the participants. Hopefully you can understand. Thank you, sir. Please continue. Please,
yeah,
sir.
sure. Yeah, that's okay. So there are different instruments that you can use depending on the type of analytes that you are interested in. So you can use NMR, you can use GCMS, you can use ESIMS coupled with LC. You can also use something called capillary electrophoresis coupled to mass spectrometry. You can IR. There are several ways that you can deal with it, and eventually you will get the metabolite, the signature of the metabolites, which can be a chromatogram, or in case of NMR, it is the NMR spectra. And then you deconvolute the data, and so from there you do the machine learning, multivariate statistics and all. Eventually you will identify signatures that are changing, and then you find out what those signatures mean and what sort of biological pathway that are involved in. And this is where a lot of effort goes in, because in some cases, for example, in NMR, you may have direct structural information, but there are several types of molecule, for example, where NMR won't give a direct structural information. Similarly, in mass spec, you certainly do not get direct structural information. You have to then look for, the calculated formula, you have to do the fragmentation analysis, and you have to then, once you identify the compound, you would then like to find out whether this is an exogenous compound or it's an endogenous compound. If it is endogenous, then what is the biological pathway that it is originating from, whether that pathway is changing, or the enzymes involved in that pathway is changing, or there is in its expression, or there is some post-transfer modification, and several questions that you can then ask to really further delve deeper into the underlying mechanism. Okay, and once you have that, as we say, that once you have that mechanism, then you can think of taking it further, either use that signature as a biomarker or use the pathway that you just elucidated, find out whether there is a druggable target and whether you can modulate that pathway, etc. Okay, so this is how it generally goes. And just to give you an idea what sort of analytical platforms are typically used, as I said, mass spectrometry and nuclear magnetic resonance, these are most widely used technique. In fact, NMR is where metabonomics, they used to call it metabonomics earlier when it started with NMR, and this is where it started. But gradually nowadays, you see more and more application of mass spectrometry. I'll tell you the reason. The first and foremost reason is mass spectrometry is highly sensitive. NMR is typically, useful when you have high micromolar or millimolar concentration of metabolites, and there are very few metabolites that are in biological fluids and biological system where you find that kind of concentration. So if you, and as I just said, that oftentimes you will find changes that are very minuscule. So therefore, if there is a metabolite which has a lower concentration in low micromolar or nanomolar and femtomolar range, you will certainly not pick them up using NMR. So you miss a lot, really a lot, if you just do NMR. On the other hand, mass spectrometry, although it is highly sensitive, it doesn't give you direct structural information, and you typically really need a lot of processing, except for the mass spectrometry-based imaging studies. For all biological fluid analysis using ESI or CT, etc., you actually need to process these samples a lot. Okay. Then there is also another advantage that that mass spectrometry has compared to NMR. That although in most cases NMR gives direct structural information, there are types of molecules, for example, fatty acids and long chain alcohols, etc., which has repeated structural units, where you will not be able to identify or tell exactly what kind of what molecule it is. So therefore, for lipid analysis, NMR is not very useful. Mass spec is quite useful for lipid analysis. So, and then you have two types of analysis, metabolomic analysis that you have. One is untargeted, another is targeted. Untargeted is when you don't know anything or you don't want to miss out on anything. This is when you do untargeted analysis. You try to analyze and quantify and identify as many metabolites as possible. And that's when you use something like LPSIMS with TOF or Orbitrap kind of analyzer, and NMR or GCMS in scan mode. If you want to do targeted analysis, you would like to use something like triple quad or Orbitrap using selective reaction monitoring, or GCMS SIM mode, or using NMR. So with that, we would like to give you a couple of examples of application of metabolomics in identification of biomarkers and elucidation of underlying mechanisms. So in this case, we'll start with colorectal carcinogenesis. I was told that it would be beneficial for the audience if I take them through at least one example where I take them stepwise how we do this whole thing, and then I will share more examples where I will not get into the every detail. So colorectal cancer, is one of the very frequent cancers, particularly in Western world, and like several other cancers, there are multiple hit models. So that means it's not a single gene that is causing it or single mutation that is causing it. There are multiple mutations and pathways, derangement of pathways that eventually lead to the formation of the tumor. So, for example, you can start with APC beta catenin pathway, and in fact, it has been seen that at least in 40 to 80% of sporadic colorectal cancer cases, this APC mutation is observed. And in addition, you can have KRAS and other mutations at later stages. So APC mutation itself is good enough for giving a neoplastic transformation. In fact, germline APC mutation causes familial type of, so hereditary type of colorectal cancer, which is called familial adenomatous polyposis coli. Okay, so so familial adenomatous polyposis or FAP. So these kind of people who has the mutation in APC gene, they often develop tumors. They develop this cancer around 35 to 40 years of age. It varies, but approximately then, most of them. So they are actually suggested that they start undergoing repeated endoscopy to find out if they are having polyps from the age of like something like 15 years or so. And that's quite invasive and not so pleasant process. So what we are trying to do here is basically two things: is to find out whether we can there is a metabolic signature that can be non-invasive, which can be used to gauge whether there is a colorectal carcinogenesis or tumor genesis that is going on. And then if it is so, then we'll try to find out what are these early signatures related to, and therefore to find out some of the pathways that might be driving this process. Okay. So with that in mind, we started with animal model. Of course, it is difficult to get a time course sample from human samples. So we thought that there is an animal model for this particular type of situation, which is called APC mean mice. So we can take these animal models and then try to see whether whatever we see in animals, whether those hold in human samples or not. So with that in mind, so this is our animal study. So the top one, the discovery cohort, you can see that there are wild type mice which will never develop these tumors, and then APC mean mice which will have intestinal tumors. So we initially started with littermate mice. That means both wild type and APC mean were from same parent, and then we have so we basically cross the wild type mice with APC mean mice. So some of them will be wild type, some of them will be APC mean. So these are genetically much closer. And then we mix them together. So the reason we did that, we have seen that even if you take mice from different parents, so there can be subtle genetic differences, although they are just mice, but because they have different parents, there are subtle differences which can alter their metabolome. So we wanted to avoid that sort of curious association. And then we also made sure that, so we just wanted to tell you that all these mice, both wild type and mutant mice, they were kept in the same cage, because we have seen from cage to cage, the mice metabolomic signature varies, mostly due to change in microbiota. Okay, so then in the discovery phase, these were very tightly controlled, and in the validation phase, we really mixed them up. So we took them from different cages, we took them from different parents, we took mice of different ages. So we just wanted to see how robust these markers are. So this is the workflow. You take the mice and then you take the urine samples from those mice, and then you do the mass spectrometry analysis, you do the deconvolution, machine learning, and then you identify the biomarker, and then you quantitate them, because eventually what is required is a quantitative information that, okay, this is the range below which there is no tumor, that and above which there is tumor, or vice versa. Okay, and then we validated that, and then we looked into we also collected tissues, and we wanted to see whatever we are seeing in the urine, whether they are connected to what's happening in the tissue or not, and then further validate in humans. Okay, so this is just to give you a glimpse of the phenotype. As you can see, that wild type and APC mean mice didn't significantly lose the tumor bearing mice didn't significantly lose body weight till the time we monitored them, but they already developed the tumor. The reason we have to take care of the body weight is that if there is a change in body weight, that itself will change the metabolome. So metabolome is sensitive to almost anything and everything that happens to you. So therefore, this is why this is so powerful. At the same time, this is so difficult to really interpret, unless you have taken into account all kinds of changes that you have been exposed to. Then we also saw that the liver function is also not significantly different. Again, liver is one of our major metabolic organs. So therefore, if there is any significant change in liver function, then there can be changes in metabolome, which may not necessarily be directly connected to the tumor genesis. So on the right, you can see that mice had tumor, and wild type mutants had tumor, and wild type didn't have. So this is unsupervised pattern recognition, just to show you that for some reason I see the healthy ones are not seen. I think there is some problem. So the healthy ones are on the left, and the tumor bearing ones, which are the black filled circle, they are on the right. I'm not sure if you can see it, but just if you are not able to. So they segregate well according to the first principle component, which tells you that there is a distinct metabotype which is associated with the development of tumor. And then it seems the one that is closest to the healthy mice is the one that had the lowest tumor burden. And as you go away along, so towards the right, mice had more and more tumor. So that means there is something in that in the metabolome of the urine, which actually correlates with the tumor burden. Then this is supervised data mining that we did. So this is called OPLSDA, and the S-plot, as we call it. So the ones which has colored red, so those were significantly upregulated in tumor, and the ones covered in green, so they were downregulated. And just to summarize, so these are different candidate biomarkers that we found that were significantly different between the healthy and tumor-bearing mice. So many of these metabolites have already been reported before, both in mouse model and human sample. What we really found novel is these two metabolites of arginine called symmetric dimethylarginine and asymmetric dimethylarginine. Okay, so this is result of arginine methylation. We will come to the pathway shortly. So just to give you a glimpse, as you can see that these you can see here that at 4 months of age itself, the level of these metabolites are significantly different and elevated in the tumor-bearing mice, and that held well in the validation cohort as well. So that means these are very novel, progressive, and reproducible signatures, which is reflected in the urine itself. So we asked ourselves, okay, if it is seen in urine, is it really connected to what's happening in the tumor, or this is just like that, the spurious association? So then we looked into the metabolome of the tumor itself. From the tumor test tissue, we found that there are several metabolites, including dimethylarginine, that were related to aberrant methylation and which was upregulated in tumor. So that means methylation at that level of the metabolome is upregulated in tumor and is also reflected in the urine of the tumor bearing mice. Then we looked whether, okay, so now we think that, okay, so something that we are finding in the urine is connected to what's happening in the tissue. Are those genes actually changing in expression? So that's actually what it is. And you can see all these protein arginine methyl transferases. You can see them significantly up, as well as several other methylation related genes were upregulated in these tumor bearing mice. We then asked ourselves, okay, if it is really happening in this tissue, how early this is? Like, is this an early driver event or a change associated with the driver phenomena, or this is just a passenger phenomena? That means tumor has, tumor genesis has taken place, and therefore these are secondary changes. So we took something. So we looked into the protein interaction network analysis, and we saw that this aberrant methylation was significantly connected with several other pathways, particularly nucleic acid and urea cycle and polyamine metabolism, which is known to be aberrant in cancer, including colorectal cancer. So that means these metabolic derangements were coordinate. And then we looked into another model where there is a conditional knockout of a conditional knockdown of the APC gene leading to beta-catenin activation. So within 24 hours of activation of this gene in the mouse intestine, the proliferation level went up, and along with that went up the methylation related genes as well as the metabolite we just talked about. And then we asked, okay, so far so good in mice, everything is falling nicely. Is it true in humans? We have to take it with a pinch of salt if you just see, things which are so good. So, but what it seems that even in human tissues also, several of these metabolites, including the dimethylarginine, was upregulated. And as you can see here, these are 10 colorectal cancer tumor tissue and normal tissues. And you can see these metabolites are upregulated in the heat map. And also when you see the one on the right, you can see only one sample out of these 10 on each case was misclassified. Okay, so at least there is 80%, 90% accuracy in terms of classification of the tumor and normal tissue. So just to summarize, what we learned from this is that this is a very convoluted picture. Don't worry about it. So these are all different metabolic pathways that we actually map from our untargeted analysis. See, if we would have started from a targeted analysis, which a priori assumes that these are the pathways that are responsible, then it would have never been possible for us to identify this novel pathway, including involving arginine methylation, that we chanced upon and found out all the way from mouse to human. So this is the power of metabolomic, particularly untargeted metabolomic, where you can really expand and get to know that where all the changes are taking place. So just to tell you, so this is the pathway which is the nucleic acid metabolism, which of course goes up when during tumor genesis. It was also known that the amino acid metabolism changes in tumor genesis. It was also reported that the polyamine metabolism changes in tumor genesis. But what we found to be novel, that along with that, there is aberrant methylation, and as you probably know, that DNA methylation, for example, is aberrant in cancer. So this is the first report where we showed that during cancer, metabolic methylation, methylation at the level of metabolites are also aberrant, and particularly arginine methylation is something that we need to watch out for. So just to summarize, we started from a beta-catenin mediated intestinal tumor genesis model, and what we found that the signatures that we picked up could actually predict tumor genesis well before. I didn't show you the ROC curves, etc. How you can combine these, just for the sake of brevity. So you can really combine these signatures, which are a reflection of coordinate metabolic reprogramming that is taking place inside the tissue or the cells towards neoplastic transformation. That sort of signature, which involves amino acid, polyamine nucleic acid methylation, polyamine metabolism and methylation, that can give you a priori hint of an the process that the process of tumorigenesis has already set in, although you may not yet see a significant tumor. So you can tell it beforehand itself, and those signatures were actually picked up even in urine. So even the urine-based metabolic signatures could, at least in mouse model, we have shown, because we could do the longitudinal study, that they could predict tumorigenesis well before it has actually taken. So metabolomics gave us not only novel biomarkers, but also biological insights into how this aberrant methylation pattern is associated with the initial events of tumorigenesis, which is as early as 24 hours within activation of FEC, the beta-catenin. So next we'll go towards another example where I mentioned that there is this gene-environment interaction that can not only affect your health, it would also perturb the metabolism. Okay, so pollutants, for example, dioxins, we know they come from plastics and other things. So these dioxins are a major risk factors. These can be endocrine disruptors, and they are also known to promote non-alcoholic steatohepatitis. So there is actually a mouse model for TCDD induced, so that's the dioxin. So TCDD induced non-alcoholic steatohepatitis. So what we are trying to do here is to find a biomarker, and just to cut a long story short, what we found, there is some metabolite which really went down, which is M0, and there were two other metabolites which went up when the M0 went down after the treatment with TCDD. So looking at the fragmentation pattern, we identified them to be azelaic acid and esters of azelaic acid. Now, azelaic acid is something that can be there in plastics and other things. We were skeptical that whether this is real or it is coming from some plasticizer and other stuff. So we then looked into whether there is a pathway to which these azelaic acid and azelaic acid esters can be connected. In fact, there is something called carboxyesterases and CES, as we call them. So again, to cut a long story short, what we showed here, that when dioxin exposure takes place, the reactive oxygen species goes up due to AHR activation. That stimulates the non-parenchymal stellate cells, which triggers the TGF-beta signaling, eventually leading to the TGF-beta SMAD pathway and the STAT-mediated pathway, leading to repression of CES3 activity. Because of the reduction in CES3 activity, CES3 is something which hydrolyzes the esters to azelaic acid and corresponding alcohol. Because the CES3 is down, now the esters are up, and azelaic acid, which is M0, that is down. So just to again give you an example, that we started with something where we have a animal model of a particular disease, and in fact we went on to show that the involvement of CES3 and this STAT-MAD pathway in several other animal models, including high-fat, high fructose-induced non-alcoholic steatohepatitis, etc. So there is a generality in this mechanism, and this particular molecule pops up in the serum of these mice even before any significant change in the AST activity, that is the liver function test that we typically do for checking the status of the liver. Even before that changes, these chemicals go up. So this is much earlier than the liver function test, because this is associated with the molecular events that are driving the non-alcoholic steatohepatitis. Now, how am I doing as far as time is concerned? How long do I have? Because then I will really speed up, or I can take time. It's up to you. How long do I have? Hello? Hello?
Hello.
Yeah, how long do I have?
Hello, sir. May I be audible?
Yeah, you are audible. How long do I have?
You have at least 20 minutes, sir.
Oh, okay, that's enough. Okay, so I just want to use slide. Oops, slide. Just give me one moment. Okay. Okay, so so this one is regarding. So now we'll come to the realm of the personalized medicine and drug discovery and drug repurposing. So when you talk about personalized medicine, the first thing that would come to our, so there are a couple of things that you have to think of. One is you have to find the right person, and then the right intervention, and then you have to come to the duration, etc. So the right person means your diagnosis has to be right. So this is an example of a biomarker, that this is a molecule that was a novel molecule. This was not reported before, the alpha derivofluoroacyl creatine, which we found again starting from untargeted analysis. We identified it and we showed that the concentration of this particular molecule goes up in urine of lung cancer patients, right, when they are stage 1 or stage 2 also. So that's relatively early, quite early compared to several other markers that we have. Now the key here is that these are urine based analytes. So that means you can, for those who are at risk of developing lung cancer, like somebody who is a smoker, etc., they can actually take these tests, which are non-invasive tests, pretty easily and do a regular monitoring without, either otherwise typically the lung cancer, when it is too late, then you will have a problem with breathing, etc. But before that, it will only be detected by chance, serendipitously, during some other scans or X-ray, etc., etc. So therefore, this one is pretty simple. It doesn't involve any expensive imaging or any exposure to radiation. It is just a simple urine-based test that you can use, that we found, and this is based on this novel molecule. And of course, there are a couple of other molecules which further improve the accuracy of this test. Okay. So this is another one example where you can detect the right person, identify the right person for the intervention. And this is another example which right now is going on in our lab and getting further expanded to multiple centers, where we try to do differential diagnosis of DKD, diabetic kidney disease, and non-diabetic kidney. Now, several people with diabetes often have this kidney problem, develop this kidney problem. Now, when somebody comes to the doctor having a chronic diabetes and then develops kidney problem, the first hunch of the doctor is basically that, okay, this is probably due to diabetes. Now, there is no specific treatment for diabetic kidney disease. Of course, just like diabetes, it cannot be cured. So you have to just take your medications for sugar and blood pressure. You have to keep them in check. That's all. However, if that person has a kidney disease which is not due to diabetes, that person may have a specific treatment available. There are several types of other disease that are seen in at least 30 to 40% of diabetic patients. The kidney disease can be due to reasons other than diabetes. So in those cases, these patients are not getting the right treatment and they can quickly die. So although there may be a treatment, just because we are unable to identify the right person, we are do the right diagnosis, the patient may perish. So the current method for diagnosis is basically biopsy. Now, biopsy is an invasive process and the time window for doing biopsy in these patients are very narrow. So therefore, oftentimes we miss the chance before it has advanced too much. So we tried to find out if there is something where we can identify things in urine and try to say that, okay, the kidney is already malfunctioning. So with that in aim, and this malfunction either is due to diabetes or due to non-diabetes. So it will serve two purposes. One, if I am to tell that, so then I'll be able to save at least 40% people from unnecessary, 60% people from unnecessary biopsy, because around 60% of them will be found with diabetic kidney disease and biopsy will be useless in that case. It will not change the course of the therapy. On the other hand, for the 40% others, we may identify them right in time and we may be able to save them, okay, or at least prolong their life. So with that in mind, we started with doing this urine-based metabolomic analysis. So we have normal, and DW is basically diabetic without any kidney disease, and then KD means they have some sort of kidney disease, and those with kidney disease may either be simply diabetic, or it's a mixed case, or it's a pure NDKD. So just to cut a long story short, that what we found that if you have M1C, distinguishing between normal to diabetic is easy. There are many robust tests available, including HbA1c and so on and so forth. We have simple regular glucose tests as well. So that's what we were not necessarily be interested in, but we just wanted to make sure that whatever signature we are getting is not just curiously associated. It is robust enough to distinguish between any kind of condition that people may come up with. So we found that if you have these M1, M2, M3, M4, I'm not naming these molecules just because they are subject to intellectual property. Unfortunately, I can't name them right away. So these molecules, if they are down in your urine, then you probably have some sort of kidney disease. Now the question is, what sort of kidney disease you have? So if you have your M6, M7, 2 metabolites up and 3 other metabolites, M8, M9, M10 down, then it is a diabetic kidney disease. And if you have, on the other hand, M5 down along with M1, M2, M3, M4, then it is probably a non-diabetic kidney disease, in which case those are the patients that need to undergo biopsy and hopefully some sort of therapy. So give you a few more examples that, okay, now that you have identified somebody, who is having a particular condition, is there some possibility, is there metabolomics, somewhere metabolomics can help us tell, okay, if this person has a particular condition, that whether a particular therapy will work or not. Because if the therapy is not going to work, it is an unnecessary harassment, and in some cases, several of these therapies may have other types of toxic side effects as well. So you do not want to subject someone to therapy unless the therapy is really going to be useful. Here is a summary of the studies where people have shown that you can actually have several of these. For example, in case of aspirin, which is used for antiplatelet aggregation, against platelet aggregation. So in this case, in case of the poor respondents, the inosine was upregulated post-treatment. Okay, and then serotonin was also found to be upregulated in plasma of poor respondents, both at the baseline, right at the beginning. You could say, okay, so serotonin is up, therefore this may not work as well. and so forth, so on and so forth. So there are several other studies which was shown to have that kind of application. So that means metabolomics can also tell you, whether a particular person is going to respond right at the baseline, and you can stratify them accordingly. So we were, I think, right at the beginning, somebody was talking about, post-COVID scenario, etc. Here is an example where people showed that, okay, so if somebody is undergoing a therapy, is it possible to tell whether the therapy is working or not? So here is a study where they showed that tocilizumab, which is IL inhibitor, so when people are treated with tocilizumab in COVID-19, it was found that the metabolic signatures could tell us whether the metabolic the profile and therefore the underlying biochemical machinery is coming back towards normal or not. So green is towards normal, red is those pre-treatment, before treatment, and post-treatment is the saffron color. So you can see there are several metabolites which comes closer to the normal level when there is tocilizumab treatment. Okay, so that means this can also reflect whether the therapy is being effective or not. You can use it to monitor. So then question is that, okay, now that I have found the right person, can I do the right intervention? So that means if I have found what's going wrong mechanistically, is it possible to use metabolomics to identify a target and then do and find out a therapy for that? So just to give you an example, you probably already know that the associated dehydrogenase mutants are involved in several cancers, including AML and then also in glioblastoma. So what these mutants do is to convert alpha ketoglutarate into 2-hydroxyglutarate, which then goes on to change, actually inhibit a lot of alpha ketoglutarate dependent demethylases. So that changes the DNA methylation and histone methylation and so on and so forth, and promotes tumorigenesis. So using metabolomics only, it was first discovered that how actually these IDAs mutants are playing a role in promoting tumorigenesis and towards poor prognosis. So metabolomics allowed identification of 2-hydroxyglutarate and then showed that if you inhibit this 2-hydroxyglutarate formation, then the phenotype can be reversed. In fact, right now there are two chemicals, as you can see, this evosidenib and this enacidenib, targeting IDH1 and IDH2 respectively, which are in clinical trials for people who specifically have IDH1 or IDH2 driven cancer, refractory AML. Okay. Again, in diabetes, people have used metabolomics and then did the data mining. So they have identified what are the metabolites that are changing with diabetes, and then found out that combine them with enzymes or proteins that these metabolites interact with. So from that network analysis, they have further identified potential several other drugs that can be repurposed for treating diabetes along with the classical ones that we have. This is another example where people have identified these these drugs, which is, for example, this afusarative. This is used in treatment of I think cervical cancer. So they have repurposed that to target glioblastoma multiforme, again starting from metabolome. And this is another study where we have shown that metformin can obliterate, significantly inhibit the proliferative capacity of hepatocellular carcinoma. So this is study from our lab itself, and so this is mediated through CIMIC regulation and involving RNA methylation. So somebody was mentioning right at the beginning, we are interested in RNA modification. So this is a case there, and this is going to be published soon. This is another example where people have already shown that production of trimethylamine oxide, which is choline metabolite, and this involves the lysis of choline using trimethylamine lyase. So there is a Clostridium genus of the bacteria which gets upregulated in some of these people who are at elevated risk of heart disease, and it was shown that this elevation in risk of heart disease in these people are because of production of this trimethylamine oxide, where the microbial TMA lyase is involved. So after that was revealed, then people use this small molecule, as you can see, which is dimethyl butanol. So they use dimethyl butanol to inhibit the microbial TMA lyase in mouse model, and then they showed that without any significant toxicity, the atherosclerotic plaques were down. So again, starting from simple metabolomics, people went on all the way to find out a novel molecule that can potentially be a drug. So towards drug discovery. And again, this is a nice paper where David Wishart in the Nature Review Drug Discovery has shown how wide you can use metabolomics to guide your drug discovery using this TMAO story as a and just to give you, as I just mentioned, that in addition to the genes, proteins, etc., that we already have, we have more than 10 times the microbial cells. So whatever number of cells we have in our body, 10 times more microbial cells are present in our body, and their gene number is 450-fold. And of course, there are many there's of these diseases, more than 1,000 typically. So you can imagine how much biochemical repertoire they have in terms of different reaction that can be catalyzed, and some of these metabolites eventually gets reflected in our biofluids, and some of them directly interact with our endogenous machinery to affect the phenotype, our health. Okay, so this is something, if we just did genomics and transcriptomics and proteomics, we will never be able to find these metabolites or the role of these microbes in our health, which are becoming and being realized more and more important as we go. Okay, so I just gave you the example of trimethylamine oxide story, where the Clostridia was involved and this DMB was used as an inhibitor. So identifying the metabolic output of our endogenous microbe and how they interact with our cells and our the tissues, that will become very important in terms of how we maintain our health and how eventual drug discovery takes place. In fact, there are several drugs, including chemotherapy drugs, where it has been shown that depending on the activity of the gut microbiota, the drug efficacy and toxicity can totally change. So therefore, all these things can be best understood only when you include metabolomics in your workflow. So just to summarize, so on one hand, you have the endogenous metabolome, and on the other hand, you have the exogenous metabolome. They coalesce together. Starting from gene transcript to the protein to the metabolite, you have to reconstruct the whole picture, and then only you will be able to tell that how the biochemical landscape is evolving, what is the machinery that is involved in a particular pathological process, and where you can target. Okay, and then only you will be able to stratify patients and find out the ones depending on the expected efficacy of the drug and also the toxicity that may result in. So based on that, you can really choose people and choose the therapy properly to to realize the goal of personalized medicine. So with that, I'd like to thank, and if there is any question, I'll be more than happy to take. So I stop here. So you can go ahead.
Thank you.
Thank you, sir. Thank you so much for this wonderful explanation. And now I would like to request all of the FDP participants that I'm opening the session for the question and answer session. And anybody, if you have any questions regarding sir's topic, please write your question in the chat box. I'll be reading it out for you to sir, and I'm sure sir would be happy to answer that. So the session is open, the platform is open for question answer. And also who are watching from YouTube also, you can put your question in the YouTube chat box as well. We can read the question from there as well. We will take your question from there and we'll read it out for sir. Just your screen is visible. If you click the option stop share at the bottom. So there is a question from Shubhankar Bhattacharya. He's asking that can the changes in TP53 gene be reversed after mutation?
Okay. Hello.
Yes, sir. Am I audible?
Yes, yes.
Yes. There's a question. There's a question in a chat box, sir. The question is asked by Shubhankar Bhattacharya. He's asking, can the changes in TP53 gene be reversed after mutation?
Well, I mean, as far as the gene is concerned, I don't think you are going to ever reverse. Once it is mutated, it is muted. Okay. So, but what can be done is that some of the downstream effects of TP53, for example, we know the P53 is a major regulator of metabolic pathways, including glycolysis, etc. Okay. So those can be tinkered with. Yes, they can be targeted, but the gene itself, I mean, metabolite is not going to, alter anything at the level of the gene. But the function of the gene that was dysfunctional or that was aberrant, that can be addressed by alternative pathways or targeting some of the things that may be upregulated, etc. Or, if because of the mutation that you may be talking about, P53, of course, is an important gene. I'm talking in general. If because of the mutation, a particular gene product, the target gene is downregulated, you can always overexpress that. Or if that gene product was making some metabolite that was doing something, for example, you can supplement that using alternative pathways. So these are definitely possibilities, but as far as changing the gene itself, that is certainly not possible.
Thank you, sir. I think one of our faculty member, Dr. Payal Chakraborty, has a question.
It was a very nice session from you, sir. I have a very short question. Say, for example, when you have the end product of lipid peroxidation.
Yes.
When you have the lipid peroxidation with 4-hydroxynonal, so you said to using your MS mass spectro, you can quantify it. So if I like to quantify the lipid peroxidation end product, and my quantifying target can be the 4-hydroxynonal, can you tell me the how can I go for that? Means what can be the alternative technique? Means I have used a fax through a IHC staining. But other than that, how can I go for it with LC mass or your this mass spectrophotometry? How can I go for that?
Yes. So the problem with this nonanal and all these kind of aldehyde containing compounds are they are quite unstable. Okay, that's one of the challenge in detecting them. However, they can definitely be dealt with using GC with appropriate derivatization and so on and so forth, and that has been done. So you can, with appropriate derivatization, you can both use GC as well as LC.
So this goes for also nitrotyrosine for the protein nitration if I'm targeting using NT.
Yes, nitrotyrosine should be easy.
I
I mean, depends on how do you want nitrotyrosine attached to the protein or nitrotyrosine standalone.
If I want to see the macromolecular damage, in one respect I want to see the lipid peroxidation, one I want to see the protein damage. In that case,
okay,
I want to quantify with one in case nitrotyrosine, and in other case I want to use the 4-hydroxynonal as my end product.
Yeah. So to cut a long story short, if you want to see the macromolecule as such with the nitrotyrosine thing, so that you can do, so there are already, you can do a proteolytic digestion and look at the peptide containing nitrotyrosine. That is certainly there. And there is also another possibility where you can probably try to think of, where you can reduce the nitro group. Okay. So you can reduce the nitro group and then attach a fluorophore to the hydroxylamine that is generated. Okay. So that is also another possibility.
Oh.
But you don't need to do that actually. You can simply look at the nitrotyrosine containing peptide. That is
enough. Oh, thank you, sir. Thank you so much.
Thank you, ma'am. Thank you, sir. Is there any other questions in the chat box? If anybody has, you can ask, sir. Sir, right now we do not have any questions, but I would definitely like to say that it was indeed a very informative session, and I hope that all of the teachers present in this FDP have got a lot of information because you explained it with very easy elaboration, sir. Thank you so much.
Thank you.
And
thank you
our
very much.
department, you're welcome, sir. Our department has a very small token of appreciation for you for sharing your time, precious time in this FDP with us and sharing your knowledge. With your permission, I would like to show that, and we'll be sending this small certificate to you over email. So if you please accept
Thank you.
it.
Thank you.
Thank you so much, sir.
Thank
you so very much.
It was a pleasure. Thank you. Okay, so I'll be logging out now.
Sure, sir. Sure, sir. I would like to request all of the participants that to stay back for a minute. I'll be sharing the link for first day FDP right now in the chat box. The link will be open for 1 hour from now. So if it's gonna be 5:30 to till 6:30, the link will be open. So you have to answer the question, and we have 5 days FDP. Every day you will be having one link. You have to answer all the questions, and the marks you need to get at least 80% marks you need to get to assure or to get the certificate for the FDP. So please try answering all the questions and make sure you are getting 80% score. You're scoring 80% to get to claim for the certificate for FDP. Now I'm sharing the link in the chat box. All of you are requested to click on that, and you can also copy it. And also I'm sharing this link in just a moment. I'm sharing the same link in the YouTube as well. It will be open for an hour. Okay, I'll be giving it shortly. In the YouTube, I'll be giving it shortly. So request all of you to click on this, or you can copy and paste it. But the link will be open till 6:30, and every day you have to answer all the question, and you have to get 80% marks to claim for the certificate. So thank you everyone. Thank you so much, all of the faculties present over here to be with us. Tomorrow on the same time we'll be starting our FDP, and will be again coming with us. Will be again joining with an eminent speaker to discuss further. Thank you so much everyone, and have a very good evening and have a very good rest of the day. Thank you.
Thank you, sir.
Okay, so somebody has asked the FDP number. So the WhatsApp group you are in, you will see you have a FDP serial number in that PDF shared by Dr. JISU-PHARMA. She is the coordinator of the FDP. Please go through that PDF has been shared in the WhatsApp link. So there you would be able to find the serial number of your, but particularly by your name. Okay, so it is FDP JISU 2022. Then there is a number. The full number you have to write in the FDP serial number that is specific for a single participant. Hopefully it is clear to all of you. Thank you so much everyone. With all of your permission, I'm right now concluding the session. Thank you.